So our presentation is on meningitis malfunction, the effect of iron on the growth of Neisseria meningitidis. And then I'm Sienna.
I'm Savannah.
So the introduction in our original experiment. So in a previous experiment this semester, we looked at how carbon sources affected cell growth. The carbon sources aided the bacteria in reproducing by acting as energy for the bacteria. Our new experiment is loosely related to this past experiment because we are examining cell growth of bacteria when it has ample amounts of iron present. In this case, the iron is acting as the bacteria's carbon source by helping the bacteria reproduce. From this lab, we learned that for bacteria to grow, it relies on outside sources, and we wanted to further our investigation on what other types of outside sources are useful to bacteria. So in our specific case, Neisseria meningitidis.
Okay, so Neisseria meningitidis is a bacteria that is solely responsible for meningitis. There's two types, commensal and pathogenic. The commensal bacteria primarily resides in the narrow nasopharynx, and it's the pathogen of Neisseria meningitidis is exclusive to humans and can be spread through nasal and oral droplets. So iron is an essential nutrient when it comes to bacterial growth. Bacteria literally cannot grow without iron. And the concentration of free iron in the human body is limited, and proteins such as transferrin, lactoferrin, and hemoglobin usually store iron, or they transport it to other parts of the body to allow the human body to function. So with free iron being in such a limited supply, it becomes very challenging for bacteria such as meningitis to grow without iron. The figure to the left shows that the guinea pigs, they did a test on guinea pigs and they infected them with C. perfringens, and they sustained a 100% survival rate when iron was not present. But then when they added iron to the guinea pigs, they suffered a 100% mortality rate.
So our question is, does an excess of iron aid in meningitis growth, and how does this correlate to attacks of the central nervous system? So with that question, we formed the hypothesis: if reproducing with excess iron available, then the meningitis will grow rapidly and attack the central nervous system. And our prediction from that is the meningitis will have more growth when there is an excess amount of iron available and will go on to negatively affect the central nervous system. So our experimental design, so our treatments, we have the blood agar plate with the meningitis present and only 50% of an iron capsule. And then for our second treatment, we have blood agar plates with meningitis present again and 100% of an iron capsule present. So the independent variable in our experiment is the amount of iron added to the plate. So if it's either half the capsule or the entire capsule. And our dependent variable is the growth of the meningitis in millimeters. So for the materials, you're going to need 7 blood agar plates, the meningitis bacteria to swab, and iron capsules to break open onto the plates. And our control is just the 1 blood agar plate with only meningitis bacteria present, because in the blood agar plates, since it is 5% sheep's blood, it still has iron present. So it'll still have a little bit of growth, just not as much as adding more iron. And for the replicants, we are doing 3 for each treatment. So 3 for the first 2 and just 1 of the control. And then our data collection. So we are collecting data by measuring the length of the meningitis growth in millimeters, kind of like we did in that past experiment. And we are also just going to visually be looking at the plates to see the thickness of the growth. And then why we chose these methods. So we chose these methods because meningitis bacteria can grow in a blood agar plate. Like I said, blood agar plates contain 5% sheep's blood, which will have iron present. Just using the blood agar plate and the meningitis as a control, we can see how little amounts of iron will affect the growth, versus by adding the additional iron with the capsules, we can then examine the effect of adding excess iron. There we
go. So this is our potential results graph for confirming our hypothesis that excess iron will increase bacterial growth. So the first bar was the agar plates with 100% of the iron capsule broken open onto it. The second bar was 50% of the iron capsule, and then the last bar is the control. Our p-value for this one was very, very small and less than 0.05, meaning there was a statistical significant difference. And then this is our potential results rejecting our hypothesis. So this one shows the opposite, that with less iron present, the amount of growth will be higher. So with the first bar, it's significantly lower than the second bar, and the first bar has more iron present than the second bar, which would reject our hypothesis.
So for our implications, the potential findings of this experiment would simply further researchers' theory that iron is necessary for bacterial growth, especially in Neisseria meningitidis. Because meningitis cannot replicate in iron deficient circumstances and environments, researchers have noticed a mutation in the bacteria. Because the iron is being stored in iron-binding proteins such as transferrin and lactoferrin, the bacteria will evolve to have receptors that allow it to attach to these proteins and extract the iron from inside in order to grow. When this happens too much, the infection rises. So this is big because bacterial meningitis is a very serious condition. According to Johns Hopkins Medicine, death can occur within hours, and without proper treatment, some lasting effects may include brain damage, hearing loss, and permanent disabilities, which also goes to show how it has a negative impact on the central nervous system. If our hypothesis was to be supported, it would provide researchers confirmation that iron is a very necessary source to keep our body functioning, but how too much of it can be deadly. With excess iron in the body, it allows pathogen bacteria to grow and could potentially cause illnesses such as liver failure, diabetes, and meningitis. And yeah,
And that's it.
that's our references.